right. Well, thank you so much for sticking around for the last talk. Like Allison said, I'm a grad student at HSU, and this was my last semester, so I did a floristic study of algae in the intertidal. But while I was working on my master's thesis, I also did this project on the side. And we actually just finished it up, and we found some results. So first of all, when I looked at the primary literature, there wasn't too much on algae in the salt marsh. We do know that both vascular and non-vascular plants are very important primary producers in the coastal wetlands, and so because they also provide food and habitat for invertebrates and fish, and perhaps birds, as well as Matt mentioned. So in addition to primary productivity, some algae are important because they fixate atmospheric nitrogen. So cyanobacteria, or blue-green algae, they fixate this atmospheric nitrogen, and then the other plants can use it to grow, basically. And this process is intensified when there's more light in the system. And then there was a study in Massachusetts that found that increases in light penetration from removal of Spartina alterniflora and Spartina patens was shown to increase the amount of benthic algae in the salt marsh. So there was salt marsh haying, where they basically removed Spartina, and they found that there was an increase in the benthic algae. And this is very intuitive, because as the canopy layers open up, there's more light penetration, and so algae can colonize and grow because of the light availability. So the objectives of this study were as follows. First of all, we wanted to know what kind of algae were found in the Humboldt Bay salt marsh, particularly in the Mad River Slough. Second, we wanted to know how the removal of Spartina would affect the algal biomass. And finally, we wanted to see how the removal of Spartina would affect the different types of algae present in the salt marsh. So we looked at different algal functional groups, basically based on their morphological characteristics, and to see if different functional groups were more prevalent in this area. So this study was done in the Mad River Slough, and here's a map showing the study plots. This was part of a larger study investigating the timing of different mowing treatments, and this study was done over 2 years. In this map, you can see that the control is this lighter square, and then the Spartina, the summer mow is these two squares right here. And there was different Spartina covers, so this area had greater than 60%, so high density, and this area was lower density Spartina. So again, these 4 treatments, there were 15 by 15 meters. There were 2 summer mows and 2 controls, and they were placed randomly in this area. A little bit more about the treatment. The summer mow was done with a brush cutter that cut the above ground biomass and also grinded up the shallow rhizomes. And in each replicate, so in each one of those 4 squares, there were 30 samples, and each we basically sampled 14 times, beginning with pre-treatment, and this was done again over 2 years. So within each treatment, we had 30 randomly selected grid intersections, and we basically laid out transect lines, and then we had quadrats that had points selected on them, and we would look at algal percent cover within those quadrats. And all the different algae that were found were placed into functional groups, based on their morphological characteristics. So we had 4 functional groups. These were green filamentous, green sheet, diatoms, and cyanobacteria. And I'll go over each one of those groups. Excuse me. For the analysis, we looked at the mean proportion of total algal cover and functional group for each of the 60 samples. So we looked at to see if basically is there more or less algae in the control versus the restored plots. And we ran a general linear model and post hoc tests for the total cover and also the cover by functional groups by time and treatment. And then we also looked at different time intervals, including pre-treatment and seasonal peaks. So for the results, we found 4 algal species in the green filamentous group. So this group was the most abundant. So there's a lot of green filamentous algae out there. And I'll show pictures of each one of these. We also found a green sheet alga. It's called ulva. And we also found diatoms and cyanobacteria, but we did not identify these to species. So here's some pictures of the different algae we found. This is Chaetomorpha linum, and you can see that it looks like green hair. Chaetomorpha refers to hair. You can see that it's these filaments and Rhizoclonium, riparium is another green filamentous. It kind of forms this kind of thinner layer of filaments. Cladophora columbiana forms pincushion. It's like a pincushion alga. Vaucheria longicaudis, this is a really common one. You can see that it's found all over the mud flats. And it's really interesting because it's cenocytic, which means it doesn't have cross walls within its cells. And here's some reproductive characteristics of it. And then ulva species. So there's different species of ulva. And they were found a little bit lower than where we were, so I didn't identify them to species. For diatoms, we found areas that were just on the mud. We could see this like brown golden color, and when I took samples of these and looked at them under the microscope, I found that there were these diatoms right on the mud. So, and again, there's quite a diversity of diatoms that we didn't identify. And the cyanobacteria form these, they're basically these mats that are about 1 centimeter thick that are darker in color compared to the mud. And here's a picture of it under the microscope. So this is what we found. So overall, this is from the pre-treatment in 2008, and then the final sampling was done in 2010. So time is on the x-axis, and percent cover of algae is on the y-axis. And we found that there were seasonal peaks in total algal cover right around May and April. So right when there's a lot of light availability, there's seasonal, the algae seem to grow quite well, and then they potentially get grazed down by invertebrates or other creatures. So the blue line is showing the control, and the restored is showing, or is the red line, and these are standard errors. So you can see that the restored peaks are both significantly higher than the control, even though the control, there's peaks as well in algal growth. So then we looked at the functional group over time to see if there was a difference between the control and the restored. And what we found was that in the control, the filamentous algae dominate the the cover. Meanwhile, in the restored treatment, there was not only filamentous, but there was also cyanobacteria and diatoms. So the first year, we're seeing diatoms and filamentous algae were growing. They both peaked in the spring. And then the second year, we saw a peak of filamentous algae, followed by a succession of cyanobacteria. And then finally, we looked at the total cover by selected time intervals. So we picked out those peaks, those specific peaks in April and May, and we we wanted to compare the control versus the restored. So these are the two treatments, the control and the restored, and we have 5 time steps instead of using all 14 that we had. So the greens are showing springtime 2009 and then 2010, and these orange colors are showing the summers, and then this was the pre-treatment. So again, there's this seasonal peak that's found both in the control and the restored, but we found that the restored was significantly higher than the treatment, and we ran the 2-way GLM with time and treatment, and we found that p-value was significant. So in conclusion, we found species of green filamentous as well as sheet algae, and as well as diatoms and cyanobacteria in these mudflats. Our results suggest that the invasion of Spartina densiflora lowers algal diversity and biomass in the Humboldt Bay salt marsh. We also found that green filamentous algae were the most abundant in both the restored and the control plots. And finally, we found somewhat of a successional pattern with the algal functional groups. So the succession was found at the 2 seasonal peaks, and also with the cyanobacteria kind of taking over at that second summer. Okay, and with that, I'd like to thank my field assistant, Bethany Bayback, Frank Shaughnessy for help with algae identification, and funding and mowing treatment provided by the U.S. Fish and Wildlife Service. And I'll take any questions.